Glycolysis and gluconeogenesis pathways by Alexis Porterfield. Glycolysis and gluconeogenesis are both pathways of glucose metabolism, but they are different. Glycolysis is the breakdown of glucose, and glyconeogenesis is the synthesis of glucose. It is important to know that glucose holds a very important job in living organisms. It is very high in potential energy, and that makes it perfect for a source of fuel for cellular metabolism. So glucose is sugar, and the body's cells need glucose to pull energy from and to perform all life's essential tasks. And cells do this by the pathway of metabolism called glycolysis. This breaking down process of glucose is an anaerobic mechanism because it does not require oxygen. Glycolysis happens in the cytoplasm of the cell, and glucose can come into the cell by glucose transporter proteins, also known as GLUT proteins. The diagram is showing the large glucose molecules cannot get into the cell, but through facilitated diffusion, with the help of those transporter proteins for the glucose, so it can enter the cell. So glycolysis starts with one glucose molecule, and by the end of the whole process, that one large molecule will be split up into two pyruvate molecules, which are smaller molecules. Pyruvate is used to make energy in the cell because glucose cannot be used as itself. It must be broken down. So glycolysis is done in two phases, where the first phase, the glucose molecule takes the energy that's already in the cell, the ATP, to be able to break it down into those two pyruvates. And then in the second phase is actually where the cell takes the energy and breaks it down to form those two pyruvates that can be used or stored in the cell as ATP or NADH. So in this picture, the diagram right here, it's showing this is your glucose molecule. Notice how large it is, and the cell can't use it as this size. So it has to go through glycolysis, which is a number of steps, about 10 steps of different enzymes catalyzing the breakdown and reactions, and the end product is these two pyruvate molecules that can then be used to make NADH or ATP. So the first phase of glycolysis, phase one, is the energy requiring phase. It has about 5 steps that are regulated by enzymes that we need to recognize. So the following, follow me in the diagram, and we'll start over here with glucose. The first step is the rate limiting enzyme, hexokinase, right here. This takes place, or takes one ATP molecule to produce that glucose 6-phosphate right here. So it takes the hexokinase to react with that. It needs that ATP to drive that to get your glucose 6-phosphate. And then your step 2, we move over using this isomerase, phosphoglucose, to make the fructose 6-phosphate right here. Notice we changed from glucose to fructose. And then it's called an isomerase because it catalyzes the change from that phosphoglucose to that phosphofructose. Step 3 here uses this phosphofructose kinase, which is very important. We're going to be talking about it later on when we start talking about activators. And it takes another ATP molecule for that enzyme to work and make that fructose 1,6-bisulphosphate. So reverting to my bullet points up here, there's the 5 processes, but the rate limiting enzymes, that's what I want you to remember right here. And that refers to rate limiting enzyme refers to the concentrations of those enzymes in the cytoplasm. So for example, if that, and I'm going to call it PFK1, this one right here, PFK1, it's used in step 3 to regulate the amount found. So when those levels of the ADH or ADP are high, the enzyme is more active. So when there is less ADP, but there's more ATP, the enzyme is less active. So that's what it means for the rate limiting. It depends on how much ATP is available or how much ADP is there. And it might sound weird thinking about it, but if there's less activation when there's ATP, because of the higher concentration, but it makes sense at the biochemical level because the ATP is the end product of glycolysis. So if there's a lot of that ATP, then why would the cell need to produce more at the moment? It doesn't. So in a way, it's inhibiting the pathway. Okay, so then we're going to move on to step 4 right here, using the fructose bisulphosphate aldose. And this is it cleaves that fructose 1,6-bisphosphate into those 2 isomers that you see over here. This one is dihydroxyacetone phosphate, and this one down here is glyceraldehyde 3-phosphate. And this moves right on into step 5, where it uses this isomer to change this to this so that it can be used further on in the glycolysis steps. Okay, and just to look back to understand and remember, we used 2 ATPs in glycolysis to perform the first phase of glycolysis. And then in phase 2 of glycolysis, this is the energy releasing phase, and we can call this over here step 6. And step 6 starts by oxidizing that sugar from that step 5. So this is from step 5. It starts to oxidize it, and it needs it's producing that NADH by the electron carrier, the NAD+. And it gains that second phosphate group, making the 1,3-bisphosphoglycerate seen at the bottom on the right corner of the diagram over here. And if there isn't enough of that NAD+ available, then it could shut down the whole glycolysis from finishing. And it's important to understand that NADH must oxidize back to that NAD+ for the process to continue. If there's not enough oxygen in the environment, this pathway moves to fermentation. So in the diagram, we have enough NADH and we're in an environment with the oxygen, so we can continue on in this phase. Step 7 is going to be right here. Sorry, I didn't number them. This is catalyzed by that phosphoglycerate kinase, and we see the ADP gains phosphate in the phosphorylation, and we see the production of ATP here. So remember, we said that this was an energy releasing phase, so we're producing that energy. We've already produced that NADH. We're producing the ATP as well. So then as we move on down, in step 8, we're using this phosphoglycerate mutase enzyme. It's to move the phosphate group to produce that 2-phosphoglycerate, pushing forward into that step 9, right over here, where we use that enzyme enolase, and it catalyzes the reaction, causing that 2-phosphoglycerate to lose a water as a dehydration reaction. That makes the double bond seen in the seen here and seen here, and above that PPE right over here. And then finally, as we get over into here, we have step 10. The pyruvate is catalyzed by that pyruvate kinase enzyme that we see in the diagram, and this creates those ATP molecules to be sent to the mitochondria to be used in the cell for energy. So then we're going to move on to gluconeogenesis, and this is the body's way of producing glucose from pyruvate and a couple of other carbon compounds that are found in the body, and this is due to the depletion of glucose for some reasons. A reason can be fasting or a major exercise workout. So those three carbon compounds used in this gluconeogenesis can be lactate, glycerol, pyruvate, and some amino acids. And this happens in the liver mostly, but it can happen in the renal cortex of the kidneys, as well as in the epithelial cells in the small intestine. The glucose made by the gluconeogenesis is put into the bloodstream to be used in those tissues that are starving for glucose. So after a heavy exercise, your muscles, they produce lactate, and lactate will make its way to the liver to be converted into glucose by the way of gluconeogenesis, and then the glucose will make its way back to the muscles to be made into that glycogen. And this movement is called the Cori cycle, but we're not going to get too deep into that. And it shows it right here. So you've used your muscles are producing that lactate, and it can't use it, so it has to transport it through the blood into the liver. The liver then can use gluconeogenesis to take that lactate, turn it into pyruvate. That pyruvate is then flows through that gluconeogenesis reactions, and then you get your ending glucose, and then that glucose can then be used back at those muscles to produce the ATP that we need. But again, this happens, excuse me, gluconeogenesis happens when the body is depleted of glucose for some reason. And again, it's going from pyruvate to glucose, the opposite direction of glycolysis, and again, it's made in select locations in the body. So gluconeogenesis, it can be said to be the opposite pathway of glycolysis, but it must be understood that some of the reactions in glycolysis are irreversible, and same with gluconeogenesis. But there is a way around it, and if we recognize anything else that happens in the body, there's always a way around something, and that is using those different enzymes. So looking at this diagram, I've cut it off about halfway so I can enlarge the words so that we can see what's going on here. And notice on the left side over here, this is glycolysis going down, and like I said up here, it got cut off purposely so we can see glycolysis. Again, we know that it starts with glucose up at the top, and it works its way down to get to your pyruvates. Now, on the right side, we have gluconeogenesis. Gluconeogenesis, the arrow got cut off, but it actually goes the opposite direction. It goes up. So we can say we start with pyruvate and work our way up using these enzymes. Pyruvate reacts with this pyruvate carboxylase, okay, to get us the oxalate, and then that reacts with PEP carboxykinase to get up here to our phospholipid pyruvate, okay, and the next three steps, you might see they look similar, familiar from glycolysis. So these two enzymes here, they do the job that to take this pyruvate to get it back up to this way. But if you notice over here on glycolysis, your pyruvate kinase is what's used to go from this major molecule to those pyruvates. They're not the same, and that's what I was meaning using those different enzymes. Okay, so in glycolysis, we use the enolase, we use the phosphoglycerate mutase, and we use that phosphoglycerate kinase. Okay, as it moves its way up from pyruvate on the glyconeogenesis. On the glycolysis, it used those same ones until we get here. Then it has to use that pyruvate kinase to get to pyruvate. Now here, we're continuing on. So we were working our way up on the gluconeogenesis side, and then we were working our way down on the glycolysis. So here we have the top portion, and these pathways, we still are using the same enzymes until we get up to here. Okay, this is a big change, and this is a big change. Now, if you remember from the previous slide, I was saying I called this one PFK1, and it is that rate limiting enzyme. And this here is also a rate limiting enzyme, fructose 1,6-bisphosphatase. It's the limiting rate for glyconeogenesis, and over here, that was the PFK1 is the limiting rate limiting enzyme for glycolysis. So As we get up here, we're using the same enzyme, but and then of course up here, this is another difference. On your glycolysis, we talked about using the hexokinase to go from glucose to glucose 6-phosphate so that it can continue to be broken down. On the gluconeogenesis side, we don't use hexokinase. We use a glucose 6-phosphate. So it phosphatase to go from the phosphate to that glucose. So it's important to understand that there are a few places where these enzymes are different so that these pathways could continue on. If we didn't have these other enzymes, then the glycolysis, it could not be irreversible, and gluconeogenesis could not work its way as well. So here we're going to look at the activators for these pathways. Glycolysis activators are glucose, insulin, AMP, and fructose 2-6-bisphosphate. And this means that when any one of these are available or are active inside the bloodstream, it will trigger glycolysis to start. So if you just ate a meal and it was full of carbohydrates, your glucose levels rise, and that will trigger those cells to start making energy by glycolysis. Insulin is a hormone produced by the beta cells in your pancreas that helps glucose enter cells and controls the amount of sugar in your blood, working with glycolysis to make pyruvate. So they kind of go hand in hand, glucose and insulin. AMP is ATP in the singular form. You might it might not have registered, but it is, and that means that there's only one phosphate, so the cells recognize this and say, hey, we need to make this into ATP, and doing this by glycolysis and then sending those pyruvates to the Krebs cycle is a way that it works together with glycolysis. So if the body sees that high amount of AMP, it will shoot into glycolysis reaction. Fructose 2 and 6-biphosphate. This is an activator for that PFK1 enzyme that I was talking about, the rate-limiting enzymes. So it's big in that step 3. So if there's a lot of that, it will kick the cell into glycolysis as well. Gluconeogenesis, on the other hand, you might see something that it triggers it saying, oh, these are opposites of each other. And that's a good thing to think about, because in gluconeogenesis, if its activators are low glucose levels, glucagon, ATP, and citrate. So in gluconeogenesis, it'll be awakened to go to work in situations where there's low glucose levels in the blood. So remember when I discussed that fasting a couple of slides ago, if you're fasting for a few days or weeks, or you work out extensively, your body is using up its glucose storages, and therefore it's starting to starve for glucose. Remember, your brain needs glucose. So gluconeogenesis gets to work, and the glucagon, like insulin, is a hormone, but it's produced by the alpha cells in the pancreas compared to those beta cells that create the insulin. And when the glucose levels are low, there will be more glucagon around. And it's the opposite of insulin, of course. So insulin has the high sugar levels, makes it activated to go into glycolysis. Um, with glucagon, it's the opposite. You have low glucose levels, so the low glucose levels go hand in hand with the glucagon in the gluconeogenesis. And then ATP, it can be used to move up that gluconeogenesis pathway in the event that the body needs more glucose because the ATP is in larger quantities. So if there's high quantities of ATP, it can use those storages to convert back to pyruvate and then move up the gluconeogenesis ladder. And then citrate. If there's high quantities of citrate from that citric acid cycle, because remember, pyruvate's used there. And again, the body says, hey, we need more glucose. Then citrate will act as an inhibitor to that PFK1 enzyme, right over here with this guy. It'll keep it from going through glycolysis. So whatever activates one pathway here, it's an inhibitor for the other pathway. So just to recap, glycolysis over here on the left, gluconeogenesis on the right, and with the help of specific enzymes in both pathways, glycolysis is that cell's way of making energy to form ATP and NADH, and this reaction occurs in the liver and in the muscles. And gluconeogenesis is making sugar out of carbon compounds found in the body to get back to homeostasis due to the depletion of glucose, and this happens in the liver and kidneys. So while both these pathways are very important, glycolysis is important in producing that cellular energy, and gluconeogenesis can only produce glucose. And since these are opposing pathways, it's helpful to understand that those activators for glycolysis inhibit gluconeogenesis, and those activators for gluconeogenesis will inhibit glycolysis. Thank you.